Thank you for the opportunity to come speak at this very important forum. And one of the purposes of my discussion is really to try to drive home some personal experiences with drug development and what has worked, for me at least, in my research at University of Illinois, and how does this research with the use of dogs with cancer dovetail into hopefully successful drug development with a novel strategy. So first, conflict of interest disclosure. I'm a paid consultant, and my research is sponsored by Vanquish Oncology, and I'm also a shareholder in this company. And so to begin with, I want to kind of just give a story that's played out very nicely so far at the University of Illinois. It really is kind of a rough roadmap that has worked for us specifically. And so if we follow this roadmap, it really started with a chemist, Paul Hergenrother, whose lab really focuses on anti-cancer novel therapeutics. And he had published a paper in Nature Biotechnology in 2006, which had identified the first small molecule activator of procaspase-3, which was called PAC-1. And Paul, in that paper, he was collaborating with people that did mirroring research, and they were very, very far away. I think they were in Korea. And so he was delighted that there was someone just down about half a mile away that he could probably work with. And so we kind of met together at Noodles and Company, and we, on a little napkin, just kind of charted out what would be the pathway to help facilitate the delivery or the development of novel therapeutics from his lab. And as we began to work together in 2007, we were able to bring the most mature compounds through in vitro work and in vivo work through the College of Veterinary Medicine, in which we did quite a bit of the piloting of these cancer and anti-cancer agents in dogs with cancer. Ultimately, this was successful enough over a few years to have R01 funding that led to the development of Vanquish Oncology, which was a startup company that was established in March of 2011. We were able to continue to produce and ultimately attracted a single angel investor that provided enough funds for us to submit an IND successfully to the FDA, which has now allowed us to launch our first in human clinical trial with this small molecule activator procaspase-3 in collaboration with the University of Illinois and Johns Hopkins University. So it's been a roadmap that so far has been very productive. We recognize there's still a long way to go before we can get past phase 1, phase 2, phase 3 study designs. And just so this was shown earlier, I think by Lee, again, the whole concept is that there are many small molecules that could be originally screened. This gets whittled down to about 250 in the preclinical and even lesser as we go forward into the phase 1, phase 2, phase 3. And I think that one of the questions that we really feel strongly about is that can you have more sophisticated preclinical model systems that would improve this hit rate or this success rate? And not to reiterate a lot, but we recognize that companion animals can serve as a unique and valuable resource in drug development. About 70 million dogs in the United States, about a quarter of them will develop cancer. And there are many potential advantages, as everyone has discussed so far today, and there are also areas of potential limitation. And I think that it will be useful as these sessions go on for us to identify those potential limitations and how can we address them and actually use dogs with cancer to be really a useful tool. One of the considerations is the population that we work with too heterogeneous? I think that most of us would subscribe that being heterogeneous is not a bad thing. So if we are able to see a signal in a very heterogeneous population with naturally occurring tumors, such as dogs, then we would hope that signal would also be visible in human beings. And then certainly we want to continue to develop uniform and broad scientific awareness and acceptance. And much of that has been driven by really the COP with Lee Hellman's lab, as well as Chan Khanna, and through multiple publications in highly regarded journals that summarize the value of the dog as a preclinical model for novel drug development. So really for our experience at Illinois, I want to kind of take you through the steps that had worked for us. And we looked at essentially the different hallmarks of cancer that could potentially be targetable. And we really focused on one of those, which is the evasion of apoptosis. And if we think about historically what therapies have been used over the past several decades, they have been focused on predominantly cytotoxins. And for these cytotoxins, they're usually targeting the DNA, and so we're looking at rapidly dividing cells. But in order for these cytotoxins to be effective, they are going to induce apoptosis, and so therefore you have to have a functional apoptotic cascade in order for these cytotoxins to work. And we know that general dose intensification with these cytotoxins is not likely to yield significant improvements in therapies. So the concept that we want to take is if we look at the apoptotic cascade, there is an intrinsic and extrinsic arm of this apoptotic pathway. And most of the defects in this apoptotic pathway tend to be upstream defects. And so if you have upstream defects in this apoptotic cascade, many conventional agents that signal through either extrinsic or intrinsic pathways will never be able to cause execution of that cell. So the theory that Paul had really thought about was, could we identify and fabricate a small molecule that directly activates procaspase-3 to caspase-3 and therefore circumvents any type of blockade that is usually existent in tumor cells? And there are many potential beneficial effects of directly activating procaspase-3. One of them is that all the apoptotic signals tend to funnel through procaspase-3, so this is a common funneling pathway. The other is that many cancers have mutations that are upstream of procaspase-3, but not many cancers have actual mutations of procaspase-3. So the target would be relatively conserved and not likely to be highly mutable. And then the other thing is that procaspase-3 levels tend to be elevated in a variety of different tumor types. And so there is the potential for a preferential targeting of procaspase-3 activation for cancer. And if we look at the biochemistry of procaspase-3 itself does have enzymatic activity. It's lowly active. When it is actually cleaved to active caspase-3, there is a feed-forward mechanism in which it's like lighting a fire, lighting a match with gasoline. So there's a feed-forward mechanism that actually triggers continued apoptosis. And so this strategy was to screen for small molecules that could have the direct ability to activate procaspase-3. And Paul's lab did identify that molecule. Out of about 22,000 molecules, they identified one drug called PAC-1. PAC-1 is the active pharmacore is this O-hydroxy and acyl hydrazone ring that actually chelates labile zinc. And so this is essentially a zinc chelator, but the affinity for binding zinc falls in a very fortuitous sweet spot. So PAC-1 has the ability to bind zinc with this type of affinity, which is enough to actually labile free zinc pools, which keep procaspase-3 in an inactive form, but is not so avid to actually pull away essential zinc ions from matrix metalloproteases or other carboxypeptidase proteins. So it is fairly selective for labile zinc pools. We're able to demonstrate that as PAC-1 comes in, based upon this active pharmacore, it binds to free zinc, labile zinc, and then causes procaspase to autoactivate to caspase-3. This, with work in collaboration with Amy Palmer, we were able to demonstrate with zinc encoded genetic sensors into cells that the mechanism of action indeed is free zinc chelation. So yellow color or warm colors indicates the presence of actually zinc, and then as the cool colors, green and blue, as you add PAC-1 at different concentrations, you actually chelate that free zinc and you have a different emission spectra that is produced. And I think that there are two essential pivotal points with regards to procaspase-3 activators as a platform, with PAC-1 being the one that we're advancing forward, is that there's an inverse relationship between procaspase-3 concentrations and sensitivity to procaspase-3 activators like PAC-1. The more procaspase-3 a cell has, such as a tumor cell, the greater sensitivity that cell has towards PAC-1. And the other thing is that there is preferential overexpression of procaspase-3 in malignant cells in comparison to their normal counterparts. So this is just a linear relationship, an inverse linear relationship with sensitivity to PAC-1 as regards to procaspase-3 concentration. And this is just graphical representation that for the most part, malignant cells have overexpression of procaspase-3 in comparison to their normal counterparts. And following this identification of PAC-1, Paul and I began to work very closely together and kind of divided different aspects of our research and how we could advance this platform. Most of the work that we had done was predominantly PK, toxicology, as well as dose-finding studies in which we are doing mouse studies, conventional mouse studies. So going back to the concept of totality information, I think that, again, we are looking at increasing the totality of information and complementing the traditional preclinical mirroring studies with what we could bring forward with dogs with cancer. And so we had dog studies, both looking at research in pet dogs with cancer, and then most importantly for our combination studies, we've looked at predominantly in dogs with cancer for this. And we've looked at PAC-1, which is the parent compound, as well as PAC-1 derivatives. These derivatives are derived based upon structure-activity relationship changes that affect pharmacokinetics, biodistribution, metabolic liability. And so we really were investigating both blood-brain barrier penetrant, which is PAC-1, as well as non-blood-brain barrier penetrant analogs that had a bulky sulfonamide group labeled to it, so it prevented blood-brain barrier penetration. And just so for our initial studies, we actually looked at a compound, a PAC-1 derivative called S-PAC-1 or sulfonamide PAC-1, has a large bulky side group, so it actually prevented it from getting into the brain. We did some different types of modeling in mice that demonstrated about 0.5% of S-PAC-1 would be found in the brain compared to 99.5% found in circulation. In the case of PAC-1, which is blood-brain barrier penetrant, 30% would be found in the brain, 70% would be found in the blood. But in our initial studies, we were able to do some dose-finding studies in normal research dogs. We at first adopted a continuous rate IV infusion strategy in which we are looking for the maintenance of key concentrations for durations of time. So we were looking at different loading doses and maintenance doses over continuous rate infusion. We were able to take that data and begin to pilot it in dogs with lymphoma, in which we were actually achieving very, very high concentrations of this procaspase-3 activator in circulation. We were able to then look at serially the lymph nodes in these dogs pre and post-treatment, and we could see that there was modulation of the target. So procaspase-3 being the target, it is actually being diminished as a function of time. And then we were also able to track by advanced imaging biologic responses based upon the tumor surface areas. And so we could see here in one example here is that when S-PAC-1 is given on a weekly basis as a continuous rate infusion, there is a gradual decrease in the actual RECIST measurement of the lymph nodes in these animals, and then upon stopping therapy, a rapid rebound in the tumor burden. Although this was reasonably positive in our findings, there was still marginal single agent activity, so we weren't happy with the overall response. There was a very cumbersome delivery mechanism, so doing a constant rate infusion for 24 hours is not very pragmatic. Most of the new molecules are going to be orally delivered, and there was toxicity if we kept it for a long period of time. So the studies from dogs with cancer allowed us to say this was a no-go strategy. And so what we then decided to think about was, could we begin to exploit the synergistic potential of low-dose pulsatile PAC-1 administration? And the concept or the hypothesis was that virtually all cancer treatments are not single agent, they are in combination or with cocktails, and that this would probably be the most reasonable path of development to follow. And so what we were able to show over the past several years is that PAC-1 or procaspase-3 activators have a remarkable ability to synergize with a broad variety of different chemotherapeutic and small molecule inhibitors. So conventional cytotoxic agents, ionizing radiation, small molecule strategies all synergize with, or PAC-1 synergizes with all these type of therapies. And so the theory behind this was, as a single agent strategy, we did have modulation of the target, we did have some anti-cancer activity, but it was not clinically the most meaningful or most opportunistic way to advance this platform. We proposed that PAC-1 could actually sensitize procaspase-3 to any other conventional cytotoxin, whether it be radiation, chemotherapy, small molecule activators, or immunobiologics, in fact. And so we felt that using PAC-1 or a procaspase-3 activating platform in conjunction was the way to move forward. And so what we did was we did a lot of detailed studies of oral bioavailability in dogs, both in research in cancer-bearing dogs, and we were able to demonstrate that the AUCs of oral PAC-1, which were highly tolerable in these dogs, were very similar to the AUCs that we were achieving in mice models of osteosarcoma. So this is the AUC of the mice, this is the AUC of dogs receiving oral PAC-1, and then we did some anti-cancer activity studies in preclinical murine models looking at osteosarcoma, looking at the ability of PAC-1 to work in combination with either doxorubicin or temozolomide. And so there tends to be a fraction of these animals that seem to be long livers whenever they have the combination. This is just some pictures to show the differences in the actual macroscopic tumor burden at the end of the study. And we decided to begin to evaluate this strategy in dogs with osteosarcoma. We were looking at first the different procaspase-3 expression patterns using some immortalized cell lines in malignant osteosarcomas as well as normal canine osteoblasts. There was, again, a preferential increased expression of the target in the malignant cell type. We could also see the expression of the target in primary osteosarcomas from dogs, as well as when we looked at paired samples, there seemed to be a good correlation between samples in the primary that were very enriched, as well as the pulmonary lesions were very enriched for that same target. And what we were able to do was to begin some early studies in dogs with metastatic sarcoma, which were treated in combination with oral PAC-1 and intravenous doxorubicin. This, in general, dogs with macroscopic sarcomas, there's a less than 5% response rate. We have right now about a 20 to 30% response rate in these dogs. So we felt that this is reasonably favorable. This is just one example of a dog that had multiple metastatic macroscopic metastasis. The smaller lesions were regressing when we were treating in combination with PAC-1 and doxorubicin versus treatment with platinum agents was ineffective. And so this pulsatile therapy has been well tolerated, and it resulted in a go decision to move forward with this strategy for metastatic sarcomas. The other thing that we really wanted to exploit was the blood-brain barrier penetration of PAC-1, and we looked at synergism with TMZ. We were really trying to focus on an unmet need for human beings with glioblastoma multiforme. And so what Rachel Botham did, who's a graduate student at Paul Hergenrother's lab, was we looked at very low concentrations of PAC-1 and the ability to synergize across a range of doses of temozolomide. And the black dotted line indicates what would be considered anything above that black dotted line is synergistic in activity. And again, what we can see is that, again, PAC-1 in combination with temozolomide results in synergism and apoptosis, as well as cleavage of procaspase-3. So again, modulation of the biologic target in combination, production of cleaved PARP as well. And in collaboration with Greg Riggins' lab from Johns Hopkins, he has actually shown in a human oncosphere intracranial implant model the combination of PAC-1 and temozolomide produces really profound improvements in survival times in these mice. And what we have tried to do is we tried to pilot this as well in dogs with primary brain tumors. This is, again, very early information, but we had an 8-year-old Labrador with a large meningioma that was symptomatically managed with anticonvulsants and corticosteroids. And we treated this dog with combination oral PAC-1 at 12.5 mg per kg on these days, and then oral temozolomide on these days. It was a 28-day cycle repeated 2 times. And we were able to do, the top panel was actually pre-treatment, and the bottom panel is a post-treatment after 2 treatments of this 2 28-day cycles. And what we can see is that via 3-dimensional volumetric reconstructions, this is the volumetric reconstruction. These are just represented same slices. We could see that there was an average of a 47% reduction in macroscopic tumor burden after 2 cycles of Temodar PAC-1. Can't say if that's from Temodar or PAC-1 or the combination. Meningiomas in general are not responsive to Temodar, so we're hopeful that it's more likely PAC-1. But nonetheless, this combination pulsatile therapy is well tolerated, and so that allowed us to make a go decision to move forward with this strategy as well. And then we have some feasibility studies that are actually planned and hopefully will start this summer looking at high-grade astrocytomas. The interesting thing that we know from these astrocytomas is the higher the histologic grade, the more aggressive these gliomas are, the greater the expression of procaspase-3 is. The potential advantage of that is PAC-1 could hopefully serve to improve the outcomes in patients, whether they be dogs or human beings with the most malignant tumor types then. And so we are actually planning these 2 pilot feasibility studies which combine oral TMZ with oral PAC-1 in dogs with non-resectable intraaxial gliomas, as well as combining it with definitive radiation and oral PAC-1. And then just so in conclusion, what we know is that PAC-1 can directly activate procaspase-3 through sequestration of Leybold's pools. It readily penetrates the blood-brain barrier and has great potential to manage central nervous system tumors. PAC-1 is orally available in dogs and can be given with conventional cytotoxic agents without any type of enhancement of toxicity. And then PAC-1 shows a wide synergism with different agents and have been demonstrated to exert activity in various clinical models. And so pet dogs for this development path was critical for the realization of the toxicity and the dosing and the limitations of single agent activity. So that was very informative for us. And it was necessary to develop the tolerable dosing regimens to inform human clinical trials and provides preliminary evidence for combination studies. And this has led to actually the launch of the first in human clinical trial of PAC-1 in human beings. This was started earlier this year through 2 centers at University of Illinois Cancer Center in Chicago, as well as Johns Hopkins. It's a 2-component study. We're in component 1 right now, which is to define the maximum tolerated dose of oral PAC-1. The component 2 is actually the exact dosing scheme that we've evaluated in the pilot study in dogs with brain tumors. So again, this is hopefully all information that will help guide and inform what would be the best dosing strategy to move forward with people. And there are many people to thank for this project, but I'd be happy to take questions.
We'll hold questions until the panel discussion at the end, if that's okay. So next is Dan Gustafson from Colorado State. Talk about PK assessment, Dan.